In this video, we will discuss about the virus mechanism of Ebola virus. So, this is the let us start from here. That is extracellular variant, and that is supposed to be comes from the exterior environment. So, in order to attach or in order to uh, enter it inside the cells, it has the receptor that is HEVCR1 or commonly known as TIM1. With the help of this, it is able to uh, endocytosis. It is able to perform endocytosis and can enter into the cytoplasm of the cell and form micropinocytosis. And after this is known as early endosome. After that, the lysosome protease, that is cathepsin B, cathepsin L, will cleave this virion, and that helps in the negative single stranded RNA, that is the genetic material that is encoded within the Ebola virus, to get transcribed or first translated into the positive single strand RNA, because positive single strand RNA can be then translated by the host ribosome and translation machinery. And this is here it is represented as negative stranded single stranded RNA, but it needs to be converted into the positive single strand RNA, and that has been done by the other components that are present within the virus. So after this, because of mRNA editing, or we can say because of the further proteases, some part of this variant will express on the cell surface, or will move into the extracellular matrix, such as SGP and VP24 and the matrix VP40 will present on the cell surface membrane, and this will helps the immune system to get activated and perform action against infected cells that are affected by Ebola virus. And this when the cleavage occurs by cathepsin B and L, the three important virals that are released in the order, like first that is VP30, VP35, and another is L, polymerase L. And these three, VP30, polymerase L, VP35, helps in conversion of negative single stranded RNA to positive single stranded RNA, and after that it can take the advantage of host uh, translation machinery, that is host ribosome and their other components. And this negative single stranded RNA, then virus replication of this negative single stranded RNA occurs after it is integrated into the host genome, and before the integration of the host genome also the translation occurs. But in order to get a stable transfection, it needs to encode, or it needs to integrate its genome with the host genome. And another thing that it will releases is that is VP24. And what's function of this VP24 is that it will prevent interferon signaling. And as we know from our previous videos, interferon signaling is one of the response, or one of the signaling response that uh, initiate the innate immunity and also the adaptive immunity. So this is about the immune response, and this is also about the immune response. So, and after this translation of this genetic material that is present in the Ebola virus, it will then move out of the move out of the cells with the help of host ESCR T mechanism. That is, uh, it is something cell sorting, something related to endosome cells, endosome sorting mechanism. More detail about this ESCRT can be find out from the reference that is available in the description. So it has been uh, after the exocytosis of this translated Ebola virus, it will then come into the extracellular matrix, and then it can affect the other cells. That's all for this video. Thank you for your time.